Yes, we are live.
Right, are we live?
Yes, we are live.
Okay, so I'm assuming we are live and that's all good. So I apologize for the little hiccup earlier. So our FB live got terminated and we have restarted it. I hope you're able to join now. I hope all is good. So can I just double check with my team? Are we ready to go live? Shall I go on?
Yes, we are
Like it?
ready. Please go
Okay,
live.
great. Thank you.
Thank you.
Thank you for the note. Okay, so once again, thank you for joining us, and a good day to you wherever you are joining us from. I, of course, most of our audience are going to be from Southeast Asia, so a good afternoon to you. I'm actually hosting this from Istanbul, so good morning here. And today, we have a really special prepared for you, Charting the Viral Human Shareome by Stephen A. James. And before we begin, I just wanted to briefly introduce this platform that this channel that we have created, DSX Live. So DSX Live has been around for some years now, and it was intended to actually connect the bioinformatics and the data science community in the region, particularly the Southeast Asia region, so that we can share our work and invite others to share their work, connect the community, and promote both research and education and services in the field of bioinformatics and data sciences. And this has been going quite well. We have had speakers in-house and from international community on a regular basis. And I think this has become even more important during this difficult times of COVID-19, and we should continue to engage with people, continue to share our work despite the difficulty in not being able to go to work, not being able to do our research in the lab. So I hope these discussions will continue to engage our minds. All right, so let me just double confirm with the team if things are still all right. Waikit, are we still okay? We're doing this FB Live through this for the first time. So just checking, Waikit, are we good to go? Everything all right? Can I get a signal? Farhan, wait, let me just. Okay, I think we are still okay. We are still live. I can see myself on Facebook. Okay, so that's good. Thank you for the signal from the team. All right. Again, apologies for the hiccup, but let's go on. So I'll now introduce our speaker. So our speaker, Stephen A. James. We fondly call him James. So he is a recent PhD graduate from Perdana University, Malaysia, and a faculty member at the Department of Biochemistry, Kaduna State University, Nigeria. He has been with us for 3 years. He was very interested to explore the world of bioinformatics, so he kind of sacrificed. It was a big sacrifice for him to leave his family and kids behind to join us in Malaysia to pursue his PhD, and I think he's done a great job, and he'll be sharing some of that work with you. And I think he has come a long way, and he will be now returning home soon once the flights resume, and be with his family, and also then embark on this journey of sharing everything he has learned and exploring more with his colleagues and students at Kaduna State University and the larger community in Nigeria. He has this passion of promoting bioinformatics and data science in his country, and I wish him all the very best in doing so. And I think it's an important effort that needs to be done, so more and more people can benefit from the wonders of bioinformatics and data sciences. All right, so, and also I'm proud to say that James is my first PhD student to graduate from Perdana University. So big congratulations to him, hearty congratulations, and we wish him all the best. And we shall begin without further ado. James, the floor is yours. Take it away. And I'll stop my video.
Thank you, Prof. Thank you for this kind introduction. Good morning, all. Good afternoon, all. Good morning, all. Wherever you are joining us, I count it a privilege, a great honor to be here on this platform to share with you some of our thoughts, some of our findings, and in relationship to shared sequences. The theme of our research, which led us to charting the viral human Shareome, is to identify the sequences of our proteome that are shared with viruses and to understand their implications in terms of function, structure, and immunological relevance. Viruses are the most abundant microbial agent in the microbiome. There are over an estimate of 9 million of them in the ocean. And there are also over 10,000 genome sequences that are available in various public repository. And also, we are well aware, it is well known all over, that viruses are obligate parasites in nature that requires the cell energy and metabolism for their obnoxious activities. Viruses also are well known as a global threat, especially in public health, as well as animal welfare. This current COVID-19 is a potent reminder of these obnoxious activities of viruses. In February 2018, precisely, the World Health Organization released the list of 9 viruses, which they mentioned as the most lethal viruses known to man, of which the list some are here presented. At the top here, we have the Ebola and Marburg from filoviruses family. We have the Zika family and also the coronaviruses, which include the SARS and also the Middle East respiratory syndrome coronaviruses, among others. Shared sequences between viral and human have been reported. This can be in the form of horizontal gene transfer homolog. And for example, viral sequences integrated into the host. The genome is not an uncommon phenomenon, and it can also be as a result of analog. That is, occurrence of this sharing is either by chance of sharing of sequences or structure or function due to convergent evolution. Thus, shared sequences between viral and human proteome could either be analog or homologs that can provide insight to the evolution of host pathogen interaction. Herein, we define shared sequences as any sequence that is 100% identical, either partial or full length, that is shared between viral and the human proteome. Viral sequences, shared sequences can also be as a result of integration, as I've said earlier. A viral sequence integrated into the host genome becomes part of the host proteome once expressed. The footprint of viruses in mammalian genome is thought to go back an estimate of tens of millions of years, in contrast to the existing estimates of few thousands. Taylor et al. Reported that several groups of mammals, including marsupial, that never colonized Africa, have had association with filoviruses. They provide a direct evidence where they show modern marsupial, like wallabies and bats, to have a fossil copies of the gene that code for filoviruses, a family of viruses that include Ebola and Marburg hemorrhagic, viruses that causes Ebola and Marburg hemorrhagic fever, among others. These viruses, like earlier mentioned, they are one of the most lethal viruses known to humans. It is also suggested that the occurrence or the maintenance of these fossil gene in this mammal is as a result, the mammal use it as a genetic defense, or there might be mere genetic scar due to persistent infection. Shared sequences can also be due to sharing due to analog between human and viruses. This is well exemplified in cytomegaloviruses, which is well known to evade immune responses by coding decoy molecules that mimic immune molecules, even to the extent of mimicking the entire length of the protein structure of the human host, especially the human major histocompatibility complex. Shared sequences have important role in viral survival, where they provide means of molecular interactions with the host. They also provide progression of life cycle. They also facilitate immune evasion. They also facilitate alteration of the host metabolic processes. A very common one also, which is well studied, is also they even hijack host cellular machineries. All these activities aid in viral pathogenicity of the human host. Specific shared sequences have been investigated through in silico studies. Kanduk et al., Luce et al., Capone et al., and David et al. Have done some work in relationship to shared sequences or sequence commonality that is being shared between viruses and human. From this chart, you can see that after searching for works that have been done in relationship to shared sequences, we find more of Kanduk et al. And Capone, which I call them as my standard, or the three musketeers that have done done good studies in relationship to shared sequences. And majorly their work view only few viral species, few viral species, and the number of protein sequences that they use is less than 100. Kanduk et al., for example, access the protein database where he reported massive peptide sharing between 30 viral protein sequences with the human proteome. And likewise, also Capone et al. Utilizes the primary protein sequences of human polio viruses, where they established some specific shared sequences also. So generally, shared sequences have been majorly studied for the major viruses like the human herpes virus, human cytomegaloviruses, which we can see here, HIV and hepatitis C virus, Zika viruses. These are some few viruses that have been studied by Kanduk et al., Luce et al., and the rest. Other motivation which made us to be more inquisitive about these shared sequences is that despite the large availability of data in the public database, much is yet to be elucidated in the aspect of shared sequences. Limited characterization of shared sequences also have not been done, especially in terms of structures and other function. Major characterization has been in relationship with immune auto reactions. The studies of shared sequences, once done, can aid us to understand the possible mechanism of viral host pathogen interaction. Will also aid us to understand the pathogenesis and evasion of the immune system. It can also give us an implication to developing suitable strategies for diagnosis procedure, for surveillance studies, for treatment, and also for vaccine development. Based on literature search, based on the gap we have identified, we come up with this big idea that there is a significant proportion of viral proteome that is expected to be evolutionarily shared between human hosts with viruses, which could have important functional, structural, and immunological implication. All these, we intend to be able to identify the shared sequences, also to be able to characterize these shared sequences, their functions, not only necessarily immunological, but any other functions. Then also to look at their structural characterization and also the immune relevance of these shared sequences. In mapping or charting the viral human shareome, we developed a workflow, which this workflow that I'm showing here is a kind of, is generic, which can be applicable to any host pathogen shareome identification. The primary source of data definitely is to retrieve data from available database. Here, majorly, I access the NCBI database, protein database in NCBI, where I downloaded all viral protein sequences. Take note of that, all viral protein sequences, and also all human protein sequences. Virtually, it means all proteins that were available as of when I downloaded these particular protein sequences, and that was in May 2018. Then we also downloaded their metadata. The next step is to process this particular data by cleaning the data, removing all unwanted protein sequences. Then generation of schemas. We developed a particular tool to generate schema at any specific defined length to construct specific schemas for our as our dictionary for our libraries, and that means we'll have in a library we'll have two dictionaries, which will be one for viruses and the other one for the humans, the human proteome. After generation of these particular schemas, we will remove all redundant by linearization process. This linearization process is to be able to identify all the metadata for each identified schemas that was generated. In each schema generated, it is generated in what we call amount format. Sorry, I don't have the format here to show you, but the format has about 3 columns. The column 1 is a peptide column, then the accession number from the source of the particular peptide that was generated, and also the position of that peptide that was also constructed. At the end of of producing the linearization, whereby we will have the unique schemas, then the viral dictionary will be used to scan the human dictionary for identical match. The identical match, whereby it is 100% identical to that of human, then that will be referred as the shared sequence. So this is the process of the mapping. 1, generation of the schemas. 2nd, removing of redundant by linearization. Then the next step thought is by scanning the viral unique schemas against that of the human. Once that is mapped, we now have the shared sequence. This shared sequence is now referred to as the viral human shareome. Thus, we define shareome as the total number of identified viral human shared sequences. The complete set of the catalog of those particular shared sequences are referred to as viral human shareome. This particular point, whereby we now have sorry, please, for that. This particular point where we now have these shared sequences, the shareome, will be the basis for every specification or consideration for our analysis. The consideration can be identifying the distribution of the schemas in the shareome. We can also carry out clustering of the shared sequences and also detection of amino acid repeats. We can also decide to look at the lineages of the viruses that are involved in the shared sequences, and also the tissue origins of the protein origins that were involved in the shared sequences, and so on. So there are various considerations that can be done depending with your area of interest. So the shareome is the basis for any further analysis. These are the various resources that are used. These are just some few resources that are being used for the analysis. Data of May 2018, which we did collected using a script provided by Entrust tailored for downloading large data sets, return 4.6 million viral protein sequences, and also for human, we had 1.2 protein sequences that were retrieved. On removal of duplicates and unwanted protein sequences, what we have at the end is 1.01, approximately 1.02 million protein sequences that were generated. And for human, we had a reduction to 304,430 protein sequences that were generated. Using our tool, which I forgot to mention here, we call it Schema Slicer, generated over 734 million kmas of peptides, kmas, sorry, for viruses, and for human, likewise, we generated over 110 million kmas that were also generated. On removal of duplicate by linearization, we had about 13.75% of these particular nanopeptides that were generated. Likewise, for human, we had about over 18 million of unique kmas or nanopeptide that were also generated. After mapping, we identified over 306 peptides that are shared between human and viruses, which represent 0.13% for viruses, initial non-redundant kmas that were generated. Likewise, it also represents 0.72 for human. These are mapped back for viruses, this particular value, which, oh, sorry. Oh, sorry for that, please. These particular numbers which we see, they very well, they are negligible amount, but this is mapped back to over 818,379 viral protein sequences. And for human, it is about 164,044 human protein sequences that these particular shared sequences originate. Looking at other considerations at this statistical distribution of the nanopeptide in the viral human Shareome, we identify the most shared sequences to be these, the most shared sequences to be this peptide here, which occur the frequency across all the viral protein sequences is this. So, and for human, it is this particular peptide, which is found in this number of protein sequences, which I cannot list here, while at least it is expected at least one protein sequences which were identified in the shared sequences to be shared. And the list is one. The list is one. The same is applicable for, the same is applicable to the human protein sequences. We also look at some consideration, which we call SSE value for the protein. This is the protein weight. This is the protein weight of a particular shared sequences that is identified over that is identified in a particular protein sequences. That is the shared sequence enrichment. Otherwise, again, which we also call shared sequence representation. This particular protein, the large Shareome protein, occurs as the most enriched or representation of the shared sequences in viruses, while in the case of human, it is the human integrated gag-pole protein of human endogenous retroviral species K with 1.6 value, while the least occurs in many viral protein sequences. The most packed viral protein sequences was from gag-pol protein with about 99%, and also the one identified in human is the human integrated large tegument protein of human herpes viral 6A with 100% occurrence for this particular sorry, let me take this off. Also, one consideration also which was done is to compare the distribution of species across the various family. From this result, you can see that the retroviruses contribute 76% of the proteins that are involved in the viral human Shareome. However, this is originated from just 2% of the entire viral species in the Shareome. While in contrast, the bacterial viruses, that is bacteriophages, from Cephaloviridea, Myoviridae, and Podoviridae, contribute the largest species if you look at the bacteriophages group of families, whereby Cephaloviridea contribute the 28% of the viral species that were identified in the Shareome. However, the protein sequences that originate from this is also around 2%. This distribution of the protein sequences in relationship to viral species, HIV-1 contribute 92% among the Retroviridae family. HIV-1 contribute 92% of the total protein sequences found in the Shareome. And while influenza from Octomyxoviridae contribute almost 6% of the entire sequences that are identified in the Shareome. Likewise, we look at, from these viruses, we look at the medically important viruses distribution in the Shareome, and we identify 5% of the viral sequences, 5% of viral species originate, I take that again, 5% of the species identified in the Shareome are those that infect human. These 5% include human viruses and non-human viruses, while only 2% infect human directly. Then, 3% affect human directly, and just 2% infect only human. And these are the values as shown here in the slides. While 95% of the remaining viruses are non-human viruses, of which, like I've earlier said, majority of bacterial viruses, with about 47% out of the 95% that infect human. Now, to understand the location of these particular protein sequences, we decided to look at the various to look at the cellular compartment component of that of the entire human protein to be able to identify their location. And from GOTAM analysis, we conducted the cellular compartment, the GOTAM for cellular component for cellular localization. And from analysis, 34% of the human protein sequences are found in the cell, while others are found in intracellular, cytoplasm, and the nucleus. And majority of these particular proteins also were also analyzed for their biological processes, and we identified about 15% to be involved in metabolism, and while others are involved in development, cell organization, nucleic acid metabolism, and among others. These are some of the biological processes that these protein sequences from human were identified. Then the molecular functions of these protein sequences that were identified, majority of them also are involved in catalytic activity. They are involved in catalytic activities. Some are also involved in signaling transduction. Others are also involved in binding and transport, of molecules across various tissue and compartments, cellular compartments. We decided to look at the network of these particular human genes. You can see, this analysis is about over 9,018 human genes that were identified by the Gene Mania to construct this particular network Shareome analysis. From for the hub analysis, we identified these particular genes as the most are the ones that are at the top of the analysis. That is the UBC NTK analysis. This analysis was produces 8,717 nodes with over 1.8 million edges. A total of 4,822 nodes genes were enriched, which are also found in this interaction. Majority of the nodes were involved in biological processes, metabolism, development, and cell communication, as earlier shown in the molecular and biological processes analysis from the GOTAM. This network analysis also showed that ubiquitin C had the highest node degree of 4,673, and it is also it is reported that to code for polyubiquitin C, which is important in maintaining the homeostasis and other normal physiological conditions of the human of humans. It also play a key role in responses associated with UV radiation, heat shock, oxidative stress, and translational impairment. Also, this particular overlap suggests that these genes may have similar function, especially from from the analysis which we did, I'm not too sure you'll be able to see some of these highlights that I will give. This is more from molecular function. There are those that are blue. The blue is from biological process, and the green is cellular compartment. I'm not too sure you will see. Why this the cyan color is an overlap of the entire genes that were identified to be involved both in cell, that were annotated for both the go terms, the three go terms, while for the ones I highlighted with the various color as well defined. So this overlap suggests that these particular proteins or genes could have similar function. And also the similarity could also suggest that analysis needs to be done to be able to ascertain, especially the relationship that could exist between these particular shared sequences and that of viruses. To look at that, I picked one particular protein sequence. Sorry, let me clean this up. I picked this particular protein sequence from a particular protein sequences, which are these all hepatitis viruses that were identified in the shared sequences were submitted to viral string database for interaction with any protein, human protein sequences to identify a specific interaction, and it returned this particular network which was generated. The TP3, that is the TP3 53 was considered as the most protein that connect the viral protein sequences which are in brown to the blue human protein sequences. This particular TP3 and PSBM7 and PSBM8 are among the protein sequences that were earlier identified to be as the hub genes. The orange nodes, like I've said earlier, denotes viral protein sequences, and the red edges are the links between the viral protein sequences to either human or within the viral protein sequences, while the gray show the interactions between the human protein sequences across also the viral protein sequences. Generally, the TP53 is a connector to both the viral protein sequences and the human protein sequences. And this particular protein also, like I mentioned earlier, is among the hub genes that were identified. And this tells us that there is an interaction between the viral protein sequences which were identified in the shared sequences with some of the hub genes, more especially the P53 and PMB7, which have a significant function, especially the TP53 is a cellular tumor antigen protein, which acts as a suppressor in many tumor types and also induces growth arrest or inhibit apoptosis, depending on the physiological condition of the cell, or depending also with the cell type. So it means from this, a lot of inferences can be drawn. From this analysis, which I've just shared as a summary, a repertoire of 100, sorry, that's supposed to be 135,606 viral shared sequences were identified. Shared sequences may be more pervasive or prevalent than expected. Also, the protein shared sequences are significantly distributed with amino acid repeats, although I didn't present the report here. Also, shared sequences were mapped to over 9,352 viral protein sequences, viral species, which are also distinguished into 178 viral families. We also identified over precisely 253 medically important viruses with their sequences, which are shared with humans. For functional analysis, to buttress and support the earlier goal terms that was done, we submitted the shared sequences to ScanProSite to be able to define the various functions of our sequences. We identify these various functions of the protein sequences. Majorly, we identify casein 2, casein kinase 2 phosphorylase site as one of the major protein sequences that were identified in the shared sequences. And also this particular protein kinase, one of the common pattern motifs that was identified is SSSE. An example of the peptide that was shown is this RSSE RLFR, which is present in EJLF2 partial glycoprotein and U2, and it's also found in U21 of herpes virus 6A, and it is shared with also the human integrated glycoprotein, which is found in human. So most of the functions of the viral, of these particular shared sequences are related to related to enzyme activity, which we have confirmed in the molecular function analysis for the goal terms, which shows that a lot of the shared sequences are catalytic. And we also see that some of them are also involved in binding and also signaling transduction. We also looked at these shared sequences, how they are they conserved, and we were able to identify some of the conserved domain, which are shown here, and collagen apolipoproteins among the top domains that were identified in these particular shared sequences. Nearly 1/3 of the shared sequences show functional relevance, matching to over 91 motifs or domain, which we identified from the ScanProSite and CDD or PFAM. Largely, the motif sites are more of phosphorylation, methylation, and also glycosylation, which are both involved in signaling transduction. We also identify a lot of these shared sequences as sites of enzymatic sites, which are associated with kinases. The shared sequences also, which we have identified, because they have these functions, then it means if we are to target these shared sequences for drug purposes, then, drug purposes, it might not be effective. Or for diagnostics purposes, there is the possibility for us to get a false positive, more especially because these shared sequences are with human. They are shared with viruses. So samples collected for analysis may likely give us a false positive. Then from structural perspective, I'm going to show only just one analysis. There are a lot of analyses which were conducted whereby we identify the relative surface accessibility of shared sequences. And we discovered that a lot of the shared sequences are exposed in comparison to their entire protein sequences. In fact, some shared sequences are totally, maybe 100% exposed. Here I present the 3D structure, the 2D structure of glycoprotein V from human herpes virus 1, which shows that the shared sequences is spread across the beta, the alpha, beta, and coil region of this particular protein. I'm not too sure if you are able to see all this area that are in purple all occupy almost the entire length of this particular protein sequences is being shared by viruses. And from our analysis, this is about from the shared sequences itself. When we consider all the shared sequences that these particular viruses present from the SSP analysis is about 100%. The localization of the shared sequences, on 3D structure show that some of the shared sequences are found in clefts. Some of them are found in clefts. For example, the HIV-1 integrase protein sequences from this arrow that you see here is found in the cleft, and the same thing for the for this particular herpes virus 1, glycoprotein D. It shows that this particular protein sequences, they are also found in the cleft, and which it may inform us that they could have some specific function in this particular position. From structural elucidation, we can gain insight on the nature of the viral architects that is involved in protein-protein interaction, and which could be a key target for drug development. And this surface accessibility also analysis can also show us the rate of the exposure of the viral molecule, and which could be also used for various interaction. But in terms of drug design or for vaccine target, we may suggest that these particular sequences should be avoided. These particular sequences that are being shared should be avoided, because one significant utility of residue being exposed is usually to present an antigenic determinant, especially epitope for viral protein in the aspect of the immunological relevant for that particular protein, such as antibodies and T cells activity. But when these particular shared sequences are similar with human or they are being mimicked by human, then it can result to autoimmune reaction. Or if they are being used as drug target, then it also means that adverse effect of that particular drug can also result once these particular shared sequences are involved in the drug design or the vaccine development. Also, analysis of the immunological relevance of our shared sequences shows that our shared sequences, majority of them were predicted to be class 1 HLA class 1 supertype restricted, with about 12 supertypes being identified in this particular, with the entire shared sequences, of which a total of 9,308 restricted epitopes were predicted as epitope, with also a number of them being promiscuous as they have multiple, which suggests they have multiple binding sites as compared, multiple binding sites. But on validating these particular shared sequences with reported records in the immune epitope database resources, we found over 161 epitopes were validated to be T cells epitopes. In total, the non-redundant shared sequences that were identified as putative HLA class 1 supertype restricted epitopes are 8,345, with a lot of them being promiscuous across multiple supertypes. And also, we also identify functional character, those that are being validated, to 11 of them to be of have immune relevance, especially with the validated ones, that is the non-redundant one among the 161 that has been identified. In the aspect of vaccine target also, or drug target, these particular epitopes should be avoided possibly, especially if they are to be included in vaccine design. In conclusion, from our study, we identify a total of 606 non-redundant shared sequences that are shared between human and viruses. We also identify that these particular shared sequences originate from over 819,000 protein sequences. We also identify that it is also originating from over 164,000 non-redundant protein sequences. The viral sequences which we identify originate from 9,352 species of 178 viral families. Like I said earlier, these sequences are more passive than expected. They also, from analysis, they show they are functional and structural and immunological relevance, which can aid us in understanding the host viral interaction. The data which we provide here can give more insight into developing suitable strategies for diagnostic procedure and surveillance and treatment, and also vaccine development. The methodology which I present here in charting viral human Shareome can also be applicable to any host pathogen, whereby it will facilitate the comparative study. From these particular shared sequences, like I studied, we were able to come up with over 40 questions to be able to understand what is all this sharing all about. It means from this particular shared sequences or viral sharing, there is more to learn. There are more things for us to learn from this comparative analysis, which was conducted. Thank you. I want to acknowledge the following people who were involved in this research, who gave me guidance, and also the platform at which I was able to conduct my research at Kaduna University School of Data Sciences, and also my funding agency. Thank you.
Thank you very much, James, for that talk. And I hope the audience found it insightful. But first of all, I think we want to apologize for the glitch. A number of you, unfortunately, because of the glitch, ended up on the wrong link. The earlier broadcast actually terminated automatically, and we had to restart. So there was a new link. Unfortunately, a number of you ended up remaining in that old link and waited. So truly apologize for that. Sorry that you were there, and we tried to very quickly rectify this and inform as many of you as possible to come and join us on this new link. So I think many of you did come, so thank you for that. Thank you for your support. So next, I think a number of you actually heard thunderstorms. So it must be raining in Malaysia, raining really badly. So it's probably about around that time of the year. And so stay indoors, stay safe. And I will just quickly check if there are any questions. All right. No questions as of now, so that's all right. This is our first time trying to broadcast direct to Facebook. Usually we do it through Zoom, so there's a learning. Maybe we're exploring something here, so hopefully maybe we've learned some lessons here. And I think in the future maybe we'll just stick to Zoom as the main platform where people come on board, and I think it'll be more interactive that way. And we can still broadcast live on Facebook, but maybe the place where people come and join would be the Zoom link. So we'll explore that the next time round. So if there are no questions, I just wanted to very quickly mention that, we do have a number of other talks lined up for the future. So do join us in the future again. If there are topics that are of interest to you, we will broadcast this, we will promote this, so you'll be aware about them. I've also shared a link on the Facebook page if you want to know more about our talks in the future and other programs that we are organizing. There's a DSxTalks link or DSX link that I shared on the comments page. So do check that out to learn about our future talks and learn about our other activities organized at the School of Data Sciences. We also have educational programs, postgraduate diploma, masters and PhD. You can check more about those on our Kaduna University website, School of Data Sciences. So if there are no talks, I want to thank our speaker, James. Thank you very much.
Asif, we have a question.
All right.
Yeah, at the chat, you can see.
All right, thank you for that. There's a question. Okay. No, I don't see any question. Can you?
It's in the Zoom chat.
Okay, so it's in the Zoom chat. Thank you for transferring. Okay, so from Mohammed, congratulations on your PhD, James. Great work. I would like to ask about if there was a fixed KMA or it varies from proteome to proteome, and does the KMA length affect the computing time? So James, did you hear that question? So I think the first question is if there was a fixed KEMA, or does it vary from proteome to proteome? I think the question is, do you recommend a fixed KEMA, or do you recommend KEMA of different lengths for different proteomes? That's the first question. Maybe you can address that.
Okay. In our own case, since we are trying to understand the workflow, the entire workflow, we stick to a specific KEMA length, which is a NIMA, but it's not for comparison. You can vary the KEMA, the KEMA length, because we discover that if you use a much bigger KEMA, you tend to restrict the number of hits and of identity. But when you reduce the KEMA length, you tend to have more shared sequences, and you tend to have more coverage to be able to capture maybe some of the functions of the shared sequences. What I would advise from what we have done, and also with comparing with other people, compare with what Lucice Kanduk did, of which they use shorter sequences and they tend to have more shared sequences. So depending with the analysis of study, I will suggest that you can vary the shared sequences for one comparative analysis. Then maybe for each proteome, you vary you vary the length. In this case, if you vary the length, then it won't be 100% identity. Although here we are talking about identity, not similarity. So I think in that case, I will suggest for a specific analysis, a specific length. For more coverage, then maybe 2 or 3 different sequence lengths can be introduced, at least to compare the results.
Right. Thank you, James. So in summary, just to summarize, I think what you're saying is that in your case, you used 9 and other lengths can be explored. And actually, the shorter the length, the more Shareome you will discover. But a lot depends on the research question that one has in mind. So yeah, you could, depending on the research question, you could explore. And I think just to clarify, James, his study was focused on identical sequences between the human and the viral proteomes, and this is not the complete repertoire of shared sequences. There would also be those that may have mutated. So similarity is another aspect that James did not explore, but that's something additionally that can be explored. Of course, that would further complicate the, not complicate, but that would further expand the analysis that needs to be done, and so one needs to carefully evaluate that pipeline before proceeding into similar shared sequences, and you need to define the parameters. But yeah, looking for identical was was a challenge itself, and especially with the big data that was involved. So coming back to this brings us to the second question from Mohammed, which is, does the KEMA length affect the compute time?
Yes, it will affect the computing time, especially the shorter the KEMA, the more space it will occupy. So that means the library, the variable that will collect each generation of the the KEMA generation, it will consume time. The mapping also will also consume time. So these particular processes, the KEMA length will affect the computational time, the generation of the KEMA and also the mapping.
Right, right. So, I mean, this is definitely another consideration, and that's why, although So besides what is the research question that you're trying to address, you also need to look at the resources that you have available. Often we want to do everything possible, but we do not have the resources. So, I mean, you got to consider all this and come to some sort of a decision on what may be the best path forward for your research question. So yeah, compute. And this was, I remember, one of the biggest challenge for James. It took him several months just to download the data, and then it also required some time to be able to analyze all those. So definitely when you're dealing with big data, you need to have those resources to be able to effectively analyze and study these sequences. All right, so I think there are no other questions. So without further ado, let me just check with the team. So can I confirm that there are no further questions?
Yes, confirm. There's no further question.
Okay.
Thank you.
So we have a confirmation from the team that there are no further questions, and we have come to the end of this session. So once again, thank you very much for your support. Thank you very much for joining us today and listening to the talk of our student James. And we wish James all the very best and all the best in Nigeria when you go back and try to spread the love of bioinformatics. Wish you all the success, and we have more talks lined up from I mean, not just from people who are well established, but also students. We would like to encourage students to come forward and present and share their work. I think that's the best way to learn. So James has led the path for this year, although this is not the first, but the first in terms of during COVID times. So thank you for leading the way, and hopefully we'll have more to follow. Thank you very much, and wish you happy day ahead. Good day ahead. Thank you.
Yeah,
Bye-bye.
Bye.
Thank you.